we'll move to the Q&A session. So, Professor Umapathy, we have some questions and hope you are comfortable. Yeah, thank you.
Sure. Yeah, please go ahead.
Yeah. So, taking up the first question, can we use the carbon quantum dots for the bioimaging using the laser spectroscopy?
Yes, but you will be doing it as a fluorescence imaging. Now, of course, these days people are using green fluorescent protein, GFPs. So carbon quantum dots can also be used because they are also very fluorescent. But you need to functionalize them to target a specific biological process or a biological cell, and therefore you can go to a particular place. You can do that, yes.
Yeah. Thank you, Professor Umapathy. And then, in line with this question, we have one more question from the chat box. Can we use a spectroscopy to identify different bacteria on skin?
Yes, we can. So we already have a patent on this. I didn't talk about it. We can actually identify up to a single bacteria level. We can say, using artificial intelligence and machine learning, about whether the bacteria is harmful or not, infectious or not. We can even study whether whether the skin lesion is fungus or bacterial infection. So we can do that.
Okay, so I can see one more question coming up in the chat box in line with this conversation. So can we get molecular structures of a tissue by laser spectroscopy besides chemical properties?
No. Structure means you're talking about morphology? No. Chemical structures you can. For example, collagen, right? You can clearly see collagen in our body, but if you want to look at the shape, that is not so easy to do because we have done, for example, invasive cancer cell imaging. And to take a single cell and do an imaging, it takes half an hour to 45 minutes to do a single cell, to look at the morphology, if we know the frequency of the chemicals in the membrane. So I would not say that Raman spectroscopy is a way to do the morphological imaging, but you can do fluorescence, you can do them to look at morphological imaging.
Okay, thank you. So we have a question around the second project that you discussed. So the question is it possible to discriminate two samples if there are multiple mutations against a single mutation?
No, unless the secondary structure changes is sufficiently large. If the mutation is only at one particular site and it doesn't affect the secondary or tertiary structures, then we may not be able to see. So what we are seeing in most of our protein studies is related to secondary structure. Primary structural changes we cannot see in proteins. We can see the tertiary structure changes as well. So unless there is a sufficiently, so multiple mutations might lead to a reasonably large number of secondary structure changes, then we can see. Sometimes even the primary structure changes could also, mutation can also lead to a change in secondary structure if there is sufficient damage in the protein or denaturation of protein, then we can identify them. So unless the mutation is very mild on a single location, it is not possible.
Okay. Coming on to the next question, how efficient is this method in comparison to conventional X-ray scanners? What are the pros and cons if we talk about?
Well, right now, X-ray scanners can do 3D imaging, but what they show you is only density. To give you an example, if you're looking at, let's say you want to differentiate between RDX and the halwa that you bought in a sweet stall, if you put both of them, if the density is the same in a suitcase, X-ray cannot tell you what it is. It'll only tell you that there is a lump which looks like an RDX or a halwa. But if you do Raman imaging, you can tell that difference. Even if the density is the same, the chemical composition will be different. So we can clearly say that it is. So that's the advantage of Raman imaging. And of course, the disadvantage of Raman imaging, we are not in a position yet to do a 3D imaging so quickly in a suitcase as the way we do in X-ray. And that takes time because you need to collect information at every millimeter around all systems, which may take maybe an hour or 2 hours, depending on the size of your suitcase or the thickness of the suitcase. So I would, or even if you look at medical side, to scan what I showed you to that 10 centimeter by 5 centimeter diameter, it takes an hour and a half, right? So there are methods to shorten the lifetime, but that we are working on. But right now it's extremely time consuming to do a large images using RAVAN.
Yeah. Thank you, Professor Umapathy. Let's move to the next question. So can we detect cancer metastasis, what all organs it might have spreaded to, based on the light scattering pattern that you have discussed?
So if the penetration length of the cancer location is less than 5 centimeter, for example, if it is near the skin surface of your body, you probably don't need a biopsy. So you can use RAVAN and identify the presence of cancer. If it is much deeper inside your body, then you might need to do a biopsy. And then if the biopsy sample is taken, then we can say whether it is cancer or not. And also we can say what grade cancer it is. And also we can say whether your radiotherapy or chemotherapy has worked or not. We can also identify whether the cancer cell has now begun to look like an invasive cell leading to cancer in other parts of body. That also can be done.
Yeah. So again, we have a question which is comprising of smaller questions, but I'll take a few of them. So how did you proceed your samples for protein analysis using Raman spectroscopy? This is the first part, Professor Umapathy. Yeah.
Right. So, let's take, for example, you can easily take a simple protein like, BSA, right? A simple protein, it's a powder. The X-ray crystallography is already well done. Everybody knows what the structure looks like. So what we do is we then run the experiment of trying to get a Raman spectrum and then trying to allot the frequencies to different parts of the protein and learn from it. So then you go on to learning about, for example, denature this protein using denaturant available in biology. And then you slowly watch how they denature them. So you learn about protein folding and unfolding. And during the process, which part of the folding happens, which is also well studied by X-ray crystallography and other fluorescence methods for folding and unfolding. We correlate those data with the Raman data. Therefore, we can identify which part of the protein we are studying when we record a spectrum. So that's how you learn about protein structure.
Thank you, Professor Umapathy. Coming on to the second part of this question, is it possible to use Raman spectroscopy to determine the toxicity of a chemical?
Toxicity, no. You can't. By just looking at a spectrum, you cannot say whether it's toxicity or not. By looking at the stability of the molecule over a period of time, by interacting with the toxic material, you can say whether something is toxic or not.
Yeah. So again, coming to a common question that might be useful for the audience, general audience. So which laser is good for biological samples if we talk then? That's
a tricky question because we have used experiment. For example, if you're looking at blood, if you're looking at hemoglobin, we use 410 nanometers, 430 nanometer wavelengths. And in some cases, we have used 632, 532, 685, 785, 785, and also 830 nanometers. So it depends on the application, depends on the substance you're studying. So you cannot easily say this is good for a particular application.
Yeah. So again, an interesting question that has come up here. So do you envision a possible use of this technology as a portable and a quick virus test kit?
We are already working on it with a friend in IISc and also with AIMS. I think it's possible within a year you may have a machine where you can tell you from your blood sample or possibly even from your swab sample whether you are infected or not, or even if you're infected, whether it is bacteria or virus, and if it is virus, which virus.
Okay. So I'll take up the next question. Can we use Raman spectroscopy for biochemical determination of marine algal tissues? And does this high salts interfere during Raman spectroscopy analysis?
No, so Raman spectroscopy only identifies chemical bonds. So salts are basically ions. You have sodium and chloride, both are ionic species, and they do not give any signal. And the most exciting part about Raman is that the water itself is not a very good Raman scatterer. So you don't get any signal from the water. And that is the reason why Raman spectroscopy is much better for medicine, because our body contains large amount of water. And infrared is not very good because water will absorb infrared. And so we can easily study as the sodium ions will not affect our studies on algae.
Thank you, Professor Umapathy. Again, an interesting question has come up from one of the students. So I have read about use of a spectroscopy to check how ripe is a fruit. Can you please elaborate about it?
Sorry, how ripe is about what?
So how ripe is a fruit?
Fruit.
Yeah.
How ripe is the fruit?
Yeah.
Yes, actually, yeah. So there is a hyperspectral imaging technology, which is actually people are using right now, which tells you how ripe, because, for example, a well-ripe fruit is very sweet. That's what people assume. Or the not ripe fruit is very sour. And by doing this imaging, they are trying to predict that. One of the things we are trying to do as well, using Raman, it's in a preliminary stage of testing, is can we actually see the sugar content, glucose content or sugar content in a fruit using our technique. And if the sugar content is large enough, and then by taking standards of extremely tasty mangoes versus not so very tasty mangoes, by comparing, we can start creating a calibration system to tell you how ripe is a fruit and how tasty is going to be.
Yeah. So thank you, Professor Umapathy. We are again getting lots of questions, and hope you are comfortable taking a few more questions. Yeah. Okay. Thank you so much. Yeah.
Another 10 minutes, 15 minutes, you can go.
Yeah. So taking up the next question, could high coherence of certain lasers might cause damage to the tissue, making it hazardous for tomography or other biological applications?
No, coherence is not an issue. Coherence is basically each photon as a wave, the phase of the wave, that is the shape of the wave, are exactly the same. So that's what we call coherence, right? And of course, because of that, the frequency is extremely pure. So that's called coherence, and that won't affect anything. What will affect is the power density. So it's like the way you're using a tube light, right? So you buy 40-watt tube light or 20-watt tube light, or you can even buy bulbs for 100 watts, the LEDs. So larger powers will damage the sample, will damage your skin. And that's why, for example, in Europe, where in summer, when they get exposed to sunlight, because of the lots of UV coming through, because of ozone hole, a lot of people who have less melanin content in their skin, particularly European and American descent, which are called Caucasians, white skin people, they get cancer, particularly skin cancer. And that is an example of how light can damage your body. So similarly, biological proteins can be damaged by high power lasers. So one has to be very careful.
Yeah. Is there any specific restriction on the biological detection, or we can detect any morphological changes using this technique?
Okay, so very good question. This is actually is the basic theory behind all these experiments. In biology, any changes in morphology is always accompanied. Even in your body, any disease that you have is always related to chemical changes in your body. Either the protein are degrading, or the membrane surface of the cell is degraded, or other components of RNA, DNA, cytoplasm, cholesterol, all of these changes is what that leads to changes in morphology or disease of an individual. So with this principle, we can always use Raman to identify any diseases.
Yeah. Again, moving on to the next question, Professor Umapathy, can we think of increasing the contrast in Raman imaging by small molecules?
So the contrast depends on the intensity of the Raman signal, right? So whether it is small molecule or a big molecule, the basic principles of Raman spectroscopy tells you the bond that you are trying to measure, it must be highly polarizable. So therefore, one has to understand the contrast will come from polarizability, the strength of polarizability, rather than the molecular size. So, for example, in functional MRI, right, they use contrasting agents to see the signals better. But in Raman, it's not the case. It happens depending on whether the sample you're looking at is highly polarizable or not.
Okay. Moving on to the next question. This is regarding the last part of your talk on using 3D Raman spectroscopy to probe for structural changes in human tissues. Is this applicable only for bones, or what all kind of tissues can be studied?
All kinds of tissues can be studied. You can look at bones, you can look at any tissues, including one of the things we are discussing about is looking at, for example, your meat that is sold in the market, right? We can see the quality of the meat. We can look at whether the meat is already too old or very young, what is supposed to be a tasty meat. So it all basically depends on whether the proteins or the composition of the structure of the meat, is it destroyed or not? So it's only accessibility as far as we are concerned, whether we can actually go in or not. It is otherwise, if there is a chemical, it can be identified. So in biology, it's always all chemical composition. Therefore, we can see.
So again, in the constraint of time, we'll take the last one question, Professor Umapathy. So I'll take this one. Can we identify 3D structures of blood cells in vivo using this technique?
Yes, we can. And this is something we have already demonstrated in the lab. Right now, we are working about trying to identify diseases based on this non-invasive in vivo detection of blood, and we are working on it.